So I am Dr. Pramod Pujari, MD in pharmacology, working at Serum Institute since 6 years, and I am handling clinical trials of the vaccines. And Cervavac vaccine was developed. Clinical trials were done by our team under Dr. Hit Sharma. So starting from preclinical phase 1 to this licensure study, what were the steps and how the data is looking like with the Cervavac as compared to the Gardasil vaccine that I am going to present. So at the outset, I would like to thank Delhi Gynec Forum for giving us opportunity to present this data. And after excellent presentation by ma'am, like that it briefed what is HPV and how it basically pathogenesis of HPV. I would like to show what is the importance of that, how the vaccines are going to prevent this. As shown here, like the microtraumas that are appearing, that are leading to the entry of HPV viruses, and then the viruses are growing, and the oncoproteins, they are causing different cancerogens and different anogenital cancers, they are caused by this HPV. Now if you look that the different steps are involved in the virus unfolding and infecting the basement membrane. All the HPV vaccines, they are based on one important component of this HPV virus, that is L1 protein. And the antibodies against this L1 protein, they are basically preventing this binding of this virus to the basement membrane, and already if it is binding virus is there, they will cause this inhibition in the conformational changes of the virus. So that's how the viruses are prevented from further infection, and they are not causing persistent infections. So antibody response with these vaccines is important parameter for their evaluation. So WHO has said that 90-70-90 is the target, 90% vaccination below 15 years. But still many of the countries in which the incidence is high, very high, but vaccination is still not there. And unfortunately India is one of them. So most important, there was no availability of affordable vaccine till date that can supply in huge quantity to these developing countries. Then vaccine reluctance was there, causing different subgroups, like they were rejecting the vaccine. Then screening also was not considered by the countries. So Serum Institute of India took that challenge of making this HPV vaccine affordable. So the driving force behind this development of this quadrivalent HPV vaccine, that is containing 6, 11, 16, 18, was to produce an effective, safe and affordable vaccine for reach out to immunization program in the private market also, and thereby reducing global and Indian incidence of cervical cancer and other HPV related diseases in boys and men also. So Serum's motto of supplying this life-saving vaccine cost effectively is supported by its huge manufacturing capacity that we have seen during the COVID times. About 90% of the Indian adult population was vaccinated by the Covishield. So we are proud that more than 25 products of Serum has got the WHO pre-qualification. And now we are coming with the different like manufacturing site at nearby Serum, so that it can increase the production of this HPV vaccine. So as brief earlier, Serum Institute again, Cervavac is a quadrivalent vaccine indicated in boys and men and girls and women 9 to 26 years. So it contains the same antigen as containing the Gardasil and aluminum hydroxide as an adjuvant. Currently it is a 2-dose vial, but we are also coming with the 1-dose containing vial also. So coming to the brief pivotal trial that we compared our vaccine with the Gardasil. So this was a partially double-blind, multi-center randomized control trial. So I will explain you why it is a partially double-blind, because we enrolled boys and girls and men and women. In that, boys and men only received Serum's vaccine because Gardasil is not indicated in. So they were like open group, while females and girls and women, they were randomized in blinded manner. They will receive either Gardasil or Cervavac. So 2 age cohorts were there, one aged 9 to 14 who received 2 doses, 0 and 6 months, while elder cohort 15 to 26 received 3 doses, 0, 2 and 6 months. So 3 arms in each cohort were there, like Cervavac only male arm, Cervavac female arm and Gardasil female arm. So this was a multi-center trial conducted 12 sites in India, and the study was guided by WHO IR, that is International Agency on Research on Cancer, Bill and Melinda Gates Foundation and DBT BIRAC. So these are the 12 sites, premier institutes which were involved in the clinical trial of this. So AIMS, PGI, then Tata Institute of Medical Sciences, all these were involved, CNCI, Kolkata, all these were the part of this study. So as it is a named phase 2/3, what we have done in phase 2, we initially enrolled 600 subjects, 100 in each arm, to see that how the vaccine is working in the terms of safety and immunogenicity. Because we have to identify the immunoassay that is correlating with the gold standard assay, that is neutralization assay, because neutralization gives that antibodies are directly neutralizing this virus, and then we can do the assay which is highly correlating with this neutralization assay. So identified the immunoassay which is to be used for the phase 3 of the subjects. Like, so 600 subjects were enrolled, safety was compared, immunogenicity was compared with the 3 different assays, and then sample size was derived, what is required for the phase 3 to produce non-inferior immune response. So after phase 2 conduction, like we assessed safety, the pain and tenderness was the most common side effect, about 41.5%, followed by induration and swelling, about 3%. Headache was the most frequently systemic adverse event, that was 14.17%, and pain in extremity was 8.33%. The adverse events reported in phase 2 part, like mild to moderate, none of the serious adverse event was reported to the vaccine. So data safety and monitoring board, which consisted of mainly oncologists, statisticians, they reviewed this data and they go ahead of the vaccine for the further development, that is phase 3. The phase 2 samples, they were tested for the immunoassay. As I said, 3 different assays were done. First is the multiplex ELISA on mesoscale discovery platform, then ELISA was done, and then pseudovariant based neutralization assay, that is gold standard assay. So we compare these 2 assays, this 1 and 2, with the PBN assay, and high correlation was found. But MSD being a high throughput assay, that means it gives the results fast, and also it tests for all 4 serotypes. In one sample we can get antibody for all serotypes. So that assay was selected for final phase 3 evaluation. So all the subjects in phase 2 were considered for again evaluation for phase 3, because till that the blinded data was there. So we calculated overall sample size required for phase 2/3 based upon the standard deviation that is derived from the immuno data, and we required about 2196 subjects. So 100 we already enrolled. Out of this, remaining 266 were enrolled in each age group. So 2196 was the target sample size. But in between COVID happened, so some subjects were lost to follow up. They were out of the town, so they could not come for vaccination or blood collection. So we approached regulatory authority that you give us the permission to increase the sample size. So about 2307, like 300 subjects were enrolled finally, and they were analyzed for the immunogenicity comparison. So primary objective of the phase 3 was to demonstrate non-inferiority of antibody responses between all the groups who are receiving Cervavac, that is 9 to 14 girls, boys, and 15 to 26 women and men, that is 3 doses. We compared with this one group only, as you see on the right side, that is women in 15 to 26 year 3 doses. And as per the WHO, you have to compare your vaccine immunoresponse with the efficacy proven population, and these immunoresponses should be non-inferior with the efficacy proven population. Coming to the results, so left side it is showing the girls in 9 to 14 age groups. The first column is the pre-vaccination geometric mean titer, and the second one is the post-vaccination geometric mean titer. The next is the comparator group in the women 15 to 26 years who received 3 doses, their pre-vaccination titer and post-vaccination titer. And finally, we compare post-vaccination titer of both these groups to see the non-inferiority response. That is, the ratio should be more than 0.5. What we observed that pre-vaccination titer was very less, and as compared after 2 doses, the post-vaccination titer was very high. That we called as geometric mean fold rise, and that was more than 1000-fold with the serums vaccine. Also with the Gardasil it was seen. But serums vaccine in 9 to 14 girls, it gives more than 1000-fold rise. And most important, non-inferiority of serums vaccine was demonstrated against Gardasil with the ratio of 0.5 against high-risk groups. That is 16-18 more important oncogenic types. Similar results were obtained for boys 9 to 14 years. Again, more than 1000-fold GMFR was seen, and non-inferiority was demonstrated with 0.5 for oncogenic types. In 15 to 26 women also, the fold rise was very high, and again the non-inferiority was met with 0.5 ratio. And similar results were in men 15 to 26 years. After 3 doses, they also showed robust immune response, and non-inferiority for oncogenic 16-18 was met. So why this robust immune response was important? As madam highlighted the one dose trial, and same is quoted here. That was the results of one dose trial published by the WHO IARC. What they have shown here, the red line you can see, that is a natural infection, naturally acquired infection. That is antibody level after that. Blue line you can see, that is after 1 dose, what is the infection. And green line after 3 doses. So after 10 years they checked that 1 dose is also giving protection, and it is 2 to 4-fold rise was there, and that is giving this prolonged protection against all serotypes. Now when we compare with this data, the robust immune response with the Cervavac, again giving the same like a 3 dose level 1000-fold rise, and that will assure the long term protection with current 2 and 3 dose schedule. Then we also assess seroconversion of this vaccine, that is subjects who are initially seronegative or who are not converted antibody, how many of the percent subjects after vaccination they converted as a positive. As you can see, it is bar touching 100% for all groups, that is serum and Gardasil, both the vaccines show 100% seroconversion post vaccination. Coming to the safety results, here we compared girls 9 to 14 years with girls, and also females with the 15 to 26 year females. So as you can see in the cohort 1, when the comparison of girls, the percentage was equal in the Gardasil and Cervavac group, while in the boys the tolerability was better. As we know that tolerability of local side effect is better for any vaccine in boys, that is seen. Similar in 15 to 26 year cohorts, girls when they compared women, when they compared with the Gardasil woman, their similar percentage was there, and in men 15 to 26 year tolerability was good. So red is for men. As you can see that side effects are less as compared to women. These are all the serious adverse events like that we have to report during the clinical trials, that if any of that causality assessment of that event should be done. So 25 events were reported which caused hospitalization mainly during the trial. But most important, none of the serious adverse event was related to the study vaccine, and none led to the study discontinuation. Because this study is planned till 3 years, so follow-up is will be continued still further, and maybe also prolonged till after that. But till 3 years we are assessing immunoresponse now. So to summarize, Cervavac has demonstrated robust antibody response. 100% seroconversion was reported across all 4 vaccine serotypes. GMFR, that is geometric mean fold rise for all HPV serotypes like types, was more than 1000-fold. Adverse events reported were predominantly mild to moderate in intensity and recovered completely. No vaccine related serious adverse event was reported, or no grade 3 solicited reactogenicity or local side effects they were reported. Overall, the incidence of adverse events with the Cervavac is within acceptable limits and in line with the product label of the comparator vaccine, that is Gardasil, and the data supports the use of Cervavac in 9 to 26 year male and female groups. as said by the madam, this was published in the Lancet Oncology editorial, and they coined this vaccine as a silver bullet, that is a simple solution for a long-standing question of HPV vaccine accessibility. So to summarize, Cervavac, that is affordable, accessible, that quality tested quadrivalent vaccine. It is India's first indigenously developed and manufactured HPV vaccine. It is a gender neutral, boys and men in 9 to 26 years it is indicated, because in boys and men they are also benefiting prevention of their cancers, and they are also giving herd immunity that prevents the infection in other also. Because IARC has also said that where the moderate vaccine coverage is there, including boys and men, it will add up the herd immunity also. It is affordable and accessible vaccine, as like the current price is very less as compared to the other vaccines. Excellent track record for assured supply, and it is in the dedicated state-of-the-art facilities this vaccine is manufactured. These are all the partners that worldwide they were involved in the development of the Cervavac. So thank you.